Thank you for visiting my poster today. This project was a part of a collaboration to investigate the impacts that feral cats have on a ringtail population on the Espíritu Santo archipelago in the Gulf of California, Mexico. Ringtails are a species of carnivore in the family Procyon, and their distribution spans much of the southwestern United States and Mexico. The population on this archipelago is very special because it is an endemic subspecies, locally called a babisuri, and is the only carnivore native to the archipelago. This archipelago has great cultural and biodiversity value, and it's home to several other endemic mammals. While the archipelago has been protected for around 30 years, feral cats have been present for around a century. Since both species are small predators that are active at night, it is possible that cats exploit similar resources as ringtails or otherwise interfere with their behaviors. Since ringtails did not evolve with cats present, it's unknown if ringtails behave adaptively to their presence and cues. In this study, we sought to describe how the cats are affecting ringtail behavior and ecology. To do this, we baited camera traps at 23 sites across the islands and recorded over 7,000 photos of ringtails at 22 sites, but only 9 photos of cats at 2 sites. Basically, we found that the ringtails are widely distributed and cats are difficult to detect. We also distributed surveys to people who frequent the islands, like park rangers and tour guides, which supplemented our cat presence data. Based on the cat presence data, we split ringtail observations into sites with and without confirmed cat presence. By looking at the distributions of their activity across time, we can see that there is no difference in peak activity between the sites with and without cats. However, the distributions have different spread. At sites without cats, ringtails are active consistently throughout the night, but at sites with cats present, ringtails are more nocturnal. This makes sense because we understand cats to be crepuscular. We also annotated the camera trap photos for any evidence of ringtail age, sex, or reproductive status. We observed evidence of reproduction through lactating females or females with dependent offspring at 13 of the 23 sites. However, we found no evidence that cat presence predicts where ringtail reproduction occurs. Lastly, we conducted a behavioral experiment. We introduced cat urine scent and evaluated differences in their visit frequency, visit duration, and other behaviors at sites. We found no difference in average visit frequency or duration, so cat scent likely does not inhibit ringtail activity at sites. However, we observed a trend where ringtails did not mark its sites until after the scent was presented. While this was not statistically significant, it could indicate that ringtails recognize cat scent and countermark territorially, as they do with other ringtail scent. In conclusion, our study found that cats likely are not limiting ringtail distribution or reproduction on the archipelago, but that ringtails may be more nocturnal at sites where cats are active. Ringtails may also behave territorially when they encounter cat cues. Further studies should examine diet overlap and agonistic interactions between these species, as they have been observed fighting over food. We want to acknowledge Rodrigo Medellin, Claudio Moreno, El Duque Adventures, our funding sources, student research assistants, and colleagues for making this project possible. You can scan this QR code to view some of our interesting ringtail observations from the field. Thanks for visiting.